Okay, hello, welcome to the third video in this series. Now this is about Unit 2 in the AS Biology spec. This is the cells unit. This is what you need to know about cells before you show up. Now it probably would be in most schools, certainly this case in ours, that you're going to get two teachers, okay, teacher 1, teacher 2, and teacher 1 will start with the biological molecules, which is the one we did in the last video, and teacher 2 will start with the cells. So these two videos here, this is for September, day 1, what we need you to know. The subsequent two videos are later material once we finish these big units. Okay, so again, I want to remind you about this GCSE to A-level progression document that's on the AQA website. Equivalents on other specs if you're watching from another school, but that's there if you want. Now make sure you're doing the transition work. The transition work for our course focuses an awful lot on the cells part, which we're going to talk about in this video. Okay, so we're going to talk about structures and functions of cells, magnification, resolution, mitosis, diffusion, osmosis, active transport, and the immune system. All of which you've covered at GCSE, all of which you should remember when you turn up on day 1 to your AS biology class. Okay, so let's dive straight into it then. Cell structures. I gave this example in the introduction video that cells are an awful lot more complicated. Right? We haven't mentioned a lot of structures so far in the GCSE. We haven't mentioned an awful lot of interesting and intricate interactions between the structures in cells, how things work. We've said things like mitochondria release energy. We've said things like ribosomes make protein. We haven't said anything about how they do that or what they link with or how they know what protein to make or so on and so forth. This at AS level is where it starts to make a bit more sense because we go into a bit more depth. Okay, so if you can come in remembering your basic animal plant cells, what you got taught at GCSE, your basic bacterial cells, remember the key difference being no nucleus, and your basic structure of a virus. Now, as I say, I'm not here to teach you this material, help you revise for your GCSEs, whatever it might be. This is a reminder of what you need to be aware of when you turn up. Okay, so there's that. And of course, it couldn't help to know where a lot of these cells come from in terms of cell differentiation and stem cells, the idea that cells specialize. You've been doing specialized cells since year 7, and we come back around to that idea of cell specialization at AS level. Okay, so that's your cell structures. This is absolutely crucial, basic math skills. Now, you might be aware that a certain percentage of GCSE and A-level science courses has to, by law, be maths, and this is a very easy way to cram some maths into A-level biology, and that's doing magnification calculations. It is unbelievably easy, right? You probably remember that's why we teach it to GCSE students, it's why we taught it to you probably in year 9, at the very beginning of your GCSE. The only trick is the units. So if we look at this triangle here, you should be familiar with this. A means the actual size of the object, I is the image size of the object, and M is the magnification. And whichever one you want to find, you cover up the other. Now, the question will have given you 2 of those to allow you to find the other one. Okay, so for example, I wanted magnification, it'd be image divided by actual. If I wanted actual, it'd be image divided by magnification. And if I wanted image, it'd be actual times magnification. Okay, so that basically is in a nutshell how that works. Now, nobody really struggles with doing that. The bit that's tricky is the unit, the micron, which you should have come across at GCSE, which is 10 to the minus 6 of a meter. Yes, you are expected to know how to use standard form. That carries on, right? Make sure you're comfortable with standard form. You will most likely be measuring in millimeters with your ruler or so on and so forth, or measuring a picture. So what you convert, that's 10 to the minus 3. So please, please, please convert one to the other before doing the calculation. You probably remember your GCSE teacher rambling on about that. We also talk about the difference between magnification and resolution, because we're going to introduce you to new and different types of microscope. That high resolution is basically sharpness. It's not magnification. I could make this picture here bigger. I could blow it up, but all I'll be doing is blowing up a blur. Unless my imaging, my camera, whatever it might be, had a higher resolution, I wouldn't see anything. Okay, and we do the definition of magnification and resolution. So going back over your microscope stuff would be really useful. Okay, we also come back to mitosis, which you would have done maybe in year 10. Mitosis, you just did a basic version, of course. The idea that when a cell comes to replicate, the DNA replicates, you get visible chromosomes, they line up, they pull apart, and the cell divides in 2. Why does this happen? So the organism can grow, repair, and go through DNA replication. If you don't remember that, go away and revise it, okay? Because we will come back to mitosis fairly soon in the new academic year. The other ones we come with your whole unit on methods of getting in and out of cells, right? So you have been introduced to diffusion, osmosis, and active transport. Diffusion, obviously, any small molecule moving down a concentration gradient to reach equilibrium. Osmosis being the passage of water through a semi-permeable membrane, okay, to balance out the concentration of something else. And then, of course, active transport, and it's time for one of Mr. Gill's patented wonderful PowerPoint animations. All right, we can see down here this little molecule, which is our energy molecule, which is ATP, which binds onto a protein and allows that protein to move and pull a substance through against the concentration gradient. So that's what ATP does. That's what active transport is, moving things through a membrane against the concentration gradient. Okay, so you need to be familiar with those and that you've done that at GCSE. Okay, and the last thing for this video is the immune system. You did a little bit of this, again, maybe towards the end of year 10, maybe the beginning of year 10, can't quite remember, but how the immune system functions. Okay, so you should know if that's a pathogen coming into somebody's body, then the little red spikes are things called antigens, and it's all about shapes on the outside of molecules. Okay, so that's a virus with antigens there. That virus will be picked up by maybe a macrophage or some sort of phagocyte and presented to a lymphocyte. This is called a helper cell. Okay, a different form of lymphocyte is then stimulated to create antibodies, like so. Okay, those antibodies are then released. They can then go over to our pathogen, do our pathogen together, immobilize them, stop them working, and allow a phagocyte to come along and consume the lot. Okay, which is a very broad overview of GCSE immunity. So if you don't remember that, go away and revise it. Okay, you also then went into vaccines. We come back to that as well. The idea being the same process works if you take away the pathogen and just put the antigen in some form. Okay, this antigen on its own will cause the lymphocytes to produce the antibodies, so they're ready, so that when the actual pathogen comes along, those antibodies are produced incredibly quickly. The pathogens are neutralized, stuck together before they can cause any sort of problem, and they're disposed of. And of course the question then is, well, how do you make sure that the antigens on their own stimulate the immune system the same way that the pathogens do? Okay, and if they do, great. If they don't, then none of that happens. Okay, and that's the problem we're dealing with, for example, in the development of the COVID vaccine. Okay, right, that is the cells topic. Plenty there for you to go away and revise. We would expect you to know those things before you turn up on day one. Next up, we'll do Unit 3, exchanges.